Hello, my name is Lauren Craig and I'm an environmental biology major at SUNY ESF. I've created this presentation to introduce Nettie Stevens by talking about the journey that she took to making her influential discovery. Additionally, I hope to emphasize the essential role that she played in defining the field of genetics. Nettie Stevens was born in 1861 in Cavendish, Vermont. However, she was raised in Westford, Massachusetts by her father and stepmother. Stevens showed an early interest in science and mathematics and ended up teaching at the high school level to fund her college education. Stevens attended Westfield State University and graduated with a Bachelor of Arts before going on to graduate school at Stanford University. After graduating from Stanford in 1900, Stevens started her PhD program at Bryn Mawr College. She remained at Bryn Mawr as a professor after graduation. Nettie Stevens had two main mentors in her scientific career. Thomas Hunt Morgan was a zoologist and geneticist that won a Nobel Prize in 1933 for a paper that Nettie Stevens collaborated with him on. Another mentor was Theodore Boveri, who Stevens studied under when studying abroad at the University of Würzburg. Boveri's research explored the role of chromosomes in heredity and provided the groundwork for Nettie Stevens' future studies. A bit of context. Stevens graduated with her PhD in 1903 from Bryn Mawr College. In the same year, another geneticist, Walter Sutton, released a paper that concluded that genetic information is found within chromosomes. In her first paper, Nettie Stevens was seeking to settle the debate that existed in the field of genetics at the time. On one side, scientists thought the accessory chromosomes came about by chromosomes disappearing due to evolution, and on the other, they thought it was involved in sex determination. To accomplish her research, she used a microscope as well as various fixing and staining techniques to analyze the testes of male insects. Since she was pioneering this field, much of the techniques she used were not written in some textbook somewhere. Because of this, before she can even start her research, she needed to develop a series of methods that would allow her to achieve the desired results. A key example of this was in the staining techniques that she used to be able to see the chromosomes on different insect species. Nettie Stevens published two papers that I would like to highlight in this presentation. The first one, Studies of Spermatogenesis, with a special reference to accessory chromosome, involved her initial discovery of sex chromosomes. The second paper attempted to determine if her initial findings could be replicated with other species. Testes of 5 different insect species were pinned in several different stains with the goal of visualizing the structure of their accessory chromosome. It was found that a combination of Fleming's strong solution, Hermann's platinoaceto-osmotic, and safranin was the best for visualizing the chromosomes in most cases. In her second paper, the chromosomes in species of Orthoptera, Coleoptera, Lepidoptera, and Hemiptera were studied. Organisms were analyzed in multiple life stages, including larval, pupa, and adult. Stevens compared organisms to those within the same order as well as those in other orders. The most important discovery came from analyzing Tenebrio molitor, or the yellow mealworm. She found that male mealworms had a significantly smaller 20th chromosome in comparison to the females. Based on the results from her first paper, Stevens concluded that this small chromosome might be a sex determinant. However, she also noted that sex may also be determined by the amount or type of chromatin in the spermatozoa. Stevens aimed to confirm her previous findings in other species. She discovered the same phenomenon of smaller accessory chromosomes in males of Coleopteran and Lepidopteran orders. Additionally, she questioned whether the X or Y chromosome was dominant. She ended up concluding that it was in fact the X chromosome. Despite all her findings, she thought that further research still needed to be done to prove or disprove that the presence of the Y chromosome was the sole determinant for the male sex, or if it was just a contributing factor. Nettie Stevens' work was incredibly influential on the field of genetics. She settled a long-lasting debate on the origins of sex determination and the function of the accessory chromosomes. Also, Stevens was the first to provide a clear link between sex determination and heredity. Nettie Stevens was not alone in her discovery. Edmund Beecher Wilson was a geneticist that independently determined that the X and Y chromosomes were linked to sex determination. Wilson is often credited with this discovery because he had previous contributions to science and a good reputation within the community. An important thing to note is that even though they were working on the same project, there was no known animosity between the two. Wilson even peer-reviewed Stevens' paper when it was initially published. Following this initial discovery, Nettie Stevens continued to contribute to the scientific literature. She discovered supernumerary chromosomes in insects and also a paired chromosome state in mosquitoes. Nettie Stevens died on May 4, 1912, at 51 years old. Despite living such a short life, Stevens contributed a great deal to the scientific community and paved the way for geneticists to come, such as Dean Hammer or Mary Lyon. Thank you for listening, and I hope you learned something.